evening, everybody. How's everybody doing today? Good. So welcome to the annual Math Science Senior Symposium. So our seniors have been working hard for the past year to get ready for this event. They have worked tirelessly. There's 16 of them. So we're going to give them 6 to 7 minutes to present the work that they've worked for the past year on. After each presenter, there will be a short Q&A if you guys have questions, and we'll keep it moving. Okay? So I hope that everybody's going to enjoy, because it's a treat.
Test. Okay. Hello everyone, my name is Mohammed Al-Zubi, and I made an autonomous RC car. So, show of hands, who uses Amazon? Okay, 1, 2, 3, 4. Okay, everyone, right? So, every time you order a package off of Amazon, an employee will take that package and they'll put it on a robot called a drive. Now these drives, what they do is, they're these orange buggies here. They'll take your package to a specific chute. Each chute leads to a specific truck, which is assigned to a specific neighborhood, which will take it right to your door. So the way these robots can navigate this grid of chutes is they'll have 2 things that they can do. They'll either have pre-simulated pathways like they do at the Hive Mind warehouse in Andover, Britain, or they'll have magnetic tape on the floor like they do at Amazon. So here in this example, they will literally have a computer called the Hive Mind that will pre-simulate all the pathways for these thousands of robots so that they never collide with each other. And here we have Bert. He's an Amazon robot. He takes employee equipment from one part of the warehouse to the other, and he just follows magnetic tape on the floor. So these methods are good and all, but the issue of them is they cost a lot of time and money to use. Because you have to pay for the magnetic tape, you have to pay for someone to lay down that tape, and in this example here, you have to wait for the computer to pre-simulate these thousands of robots, pathways. So to remove these methods, I made an autonomous RC car here, and its goals were to reach a destination in a 3x4 grid without hitting any obstacles, and the second goal was to stop at the sign for at least 5 seconds. So the way the robot can actually detect its environment is it has a webcam, a display screen which just shows me what the webcam is seeing, then a Raspberry Pi board which controls all of the components of the robot, and lastly an ultrasonic sensor which can identify obstacles on the grid. So the way that ultrasonic sensor actually can identify obstacles is what it'll do is it'll shoot out a sound wave, and when it's echoed back to it from the obstacle, it will know how far it is from the obstacle based on the speed of sound. And with regards to traffic sign detection, the way it does that is the webcam video will be sent to the Raspberry Pi. The Raspberry Pi will analyze it using OpenCV, Open Computer Vision, and it'll outline the shape it sees. So here it made an outline in green, and then it'll count the number of points of the shape it just made. So if it counts 8 points, then it knows that it's an octagon, and it knows to stop. And so let's say it counted 3 points, it would know it's a triangle, and it wouldn't stop at that sign, because its condition is that, does this shape have 8 points? Then we stop, else we do not stop. So, setup. I had the robot tested 40 times. Each time the location of the obstacles were randomized, as well as the location of the stop sign and the location of the destination. So the part of the pre-verification process was to make sure that the robot had a possible way to get to the destination, and also that the robot encounters all 4 obstacles and the stop sign, so that you won't have environments where, let's say, it only encounters one obstacle and then it can get to the destination. That's not a good testing run. So as to how the robot can actually navigate the grid, here you can see a picture of my setup in my garage. And so what it does is has an algorithm that will plan the path from the beginning. So let's say in this example, it's trying to get to A3, right? So what it'll say is, okay, let's take the simplest path. Let's go up and to the right, and it'll do that. It'll try that. So it'll go up and it encounters, obstacle 1 right here. So we say, okay, well, if we can't go up, then let's try going right, and then up. So it'll do that. It'll try going right and then up. Encounters obstacle number 2, says, okay, we cannot do that, so let's continue on this way, and up and left and up and right, and there we are at the destination. So the eye recorded, did it reach the destination, did it hit any obstacles, and did it stop at a sign. That's why I recorded for each of these 40 runs. And so results, the robot reached the destination 90% of the time, it stopped at the sign 95% of the time, and it hit obstacles only 8% of the time. So I would say the robot is fairly successful in its goals. However, there is room for improvement. So some reasoning as to why the robot is hitting obstacles is the fact that 70% of all collisions were on a turn tells us something. It tells us, okay, we can see here that when the ultrasonic sensor shoots out its sound wave, sometimes it's not reflecting back to the sensor. So it never detects the obstacle and it plows on right through. So to fix that, we can add ultrasonic sensors to the sides of the robot so we have a higher range of detection. Because imagine this is our range of detection right now. We're trying to increase it, okay? So we can add additional ultrasonic sensors, or a more effective solution would be to have one singular LiDAR sensor. What the LiDAR sensor does is, as you can see here, it shoots out a laser in a 360-degree motion so that it can detect obstacles from all angles. Now, the cost of this sensor is $500, so a cheaper yet still effective solution would be to use a radar sensor. A radar sensor, it's used in smart cars today, actually, used as parking aids, and it's half the price of the LiDAR sensor, sitting at only $250. So it definitely is more expensive than our ultrasonic sensor right here. This is only $5, but it has a much higher range of detection. So for this purpose, it's very optimal. One last thing I would like to delve into is having, instead of stop signs at intersections, having traffic lights at intersections. Because obviously having a bunch of stop signs at intersections is not going to be able to control the traffic of thousands of robots very well. So, traffic lights would be much more efficient at this, as we do in the real world, we have these. Here are my references. I would like to acknowledge my mentor, Dr. Abidin Yildirim, and my advisor, Dr. Flavonoid, and Ms. Chin for everything she does. Thank you all for coming, and
Are there any questions?
I have one.
yes?
Are there any environmental concerns that would disrupt the sensors for the LiDAR, for example, or is that, you don't need a clean room or anything?
As long as the LiDAR sensor can, in fact, I guess what you're trying to say is any environmental impacts.
Yeah.
So, the entire reason we're using the LiDAR sensor is because it's unaffected by these environmental things. So the ultrasonic sensor, the way, again, I'm going to explain again. So it has a sound wave. So if we have a bunch of noise happening, these ultrasonic sensors cannot work at full accuracy. So that's why we're getting away from these ultrasonic sensors and moving to something like a LiDAR sensor, which would be more accurate and it won't be affected by these environmental effects.
Great, thanks.
Mm-hmm. Any other questions? All
We
right.
have a mic now for questions if anyone wishes to use it.
Is that Mr. Ware?
Yeah.
Yes, Mr. Ware?
Did you you ever have multiple robots interacting, or did you think about having multiple robots?
Yes, I did think about that. However, the cost to make each of these robots was $150. The originally RC car was $100. All the components added on was $50, so $150. So I wanted to have at least two, but the RC car that I ended up buying was really pricey, so I ended up just having one singular robot. But that is something that would be need to work on in the future, how they interact with each other. Yes.
Cool.
I have a question.
Yes.
What problems did you foresee with multiple robots using only stop signs, and how did that lead you to traffic lights being a solution?
Yes, so the entire reason for me having stop signs was I never expected it to be a good traffic controller, really. I just wanted to see, can this camera identify traffic signs? I just want to see how capable is it of identifying traffic signs. So, you know, like, I stepped into the area, and now moving on to traffic lights would be the real optimal solution. That was the point of the traffic signs, of the stop signs, just to delve into it, just you know, get my toe in the water, you know.
All right, the main part.
Hello, test.
One. I saw the movie.
All right, hello, my name is Aaron Pia, and I'm here to present to you my research about using machine learning to predict the average effective areas of muon neutrinos. So there are three main things you'll need to know for really trying to understand what I'm doing here. First of which is, what is a muon neutrino? Well, a muon neutrino is a type of elementary particle that is produced when a cosmic ray of particles outside of Earth interacts with molecules in the atmosphere. This causes a chain reaction of decaying particles. So this is shown over here with the formation of a pion and antipion, which are just another set of elementary particles. They decay into several particles, most prominently being the muon neutrino. The second thing is, what is an effective area? Well, an effective area is essentially where the area where particles are physically present. So this is shown in this diagram with a bunch of particles coming out of the effective area. This right here is a detector area, which I'll explain in this slide. So the detector area is exactly what it sounds like. It's the area of the detector within the specific effective area that where local particles are able to be detected. So this right here, this diagram, is actually the detector researchers use to collect the data that I used in my model. So this right here, highlighted in red, is the Qpcr detector, which is in the IceCube Observatory, which is located in Antarctica. My research goal is to be able to predict the average effective area of specifically muon neutrinos within the energy of 2 to 10 GeVs. So GeVs are giga-electron-volts, and they're just the amount of energy the neutrino emits. And to do this with a minimal validation and training loss value of less than 1. So the loss value is essentially just a method of evaluating how well the model I'll be creating fits the data set. Moving on to my methodology, I have 4 steps. The first step is to obtain the data, then process it, then compile the model, and finally visualize the results. So the first step, obtain. I obtained all of my data from the IceCube Observatory's public database. The data consisted of 5 CSV files, each containing 2,000 entries of data, and each entry contained an energy value as well as an average effective area value. I then used a software called Jupyter Notebook to begin coding, and first thing I did was combine these 5 CSV files into one. I then took that combined CSV file, converted it into an array, so I'll be able to manipulate it. I took that array, shuffled it, and then I split it into two separate arrays. One for the input, which is the energy value, and one for the expected results, which are the average effective areas. I then took the array of expected results and then scaled it to a maximum of 10 in order to reduce the loss calculation I'll talk about in this slide. So in order to create my model, I use Keras, which is a type of machine learning library, which allows me to access a bunch of machine learning functions I'll be able to use in my code. So I created a sequential model, which just executes the layers in order, and it consisted of an input, 2 dense layers, as well as an output layer. I then compiled my model with the loss calculation of mean squared error, which just squares the difference between the model's prediction and the actual value I input into the model. I then fit the model with the input, the output, the epochs, which is just the number of times the model is run. So if I set the epoch to 1, the model will just run once. So I set that value to 500, and I have the validation split set to 0.3. So the validation split is used to prevent overfitting. So overfitting occurs when the model fits a particular set of data too well, causing it to only work for that particular set of data. So by training it with one data set and then validating it with another, I'm able to reduce overfitting. Finally, I visualize my results using the matplotlib library, which allows me to create the graphs you see here. So these graphs show a significant decrease in the first few times the model is run, and then an overall constant rate for the rest of the epochs. From these graphs, we can conclude that the minimal training loss value is approximately 0.184, and the minimal validation loss value is approximately 0.1977. So from this, we can conclude, from these values, we know that, my research goal was accomplished because both of the loss values were both less than 1. Although my research goal was accomplished, there is a limitation to this, and that is that since I got all of my data from the IceCube Observatory, that means it can only be extrapolated for that specific observatory and its method of data collection. So what this means is, as I mentioned before previously, the detector I got all of my data from, DeepCore, is located in Antarctica. So things like temperature and location could definitely affect my results. Some things I could do in the future to improve my results and improve my accuracy is use a bigger range for the energy bins. So what that means is instead of using something like 2 to 10 GeVs, I could use something like 2 to 100 or 2 to 1000. So this would allow me to extrapolate results for not only neutrinos that are formed in the atmosphere, but for neutrinos that are formed beyond Earth or even the solar system, from stars and even black holes. Also, using data from different observatories could yield accurate results. So as I mentioned earlier, temperature could play a big role in how my results were displayed. So getting data from different observatories could definitely make my results more accurate. That is all for my presentation. I'd like to acknowledge Dr. Hoya, Mr. Sproul, and Ms. Chin for helping me with my research. Thank you.
So in order to keep the program moving along, we'll do question and answers at the very end. So we want to just keep the presentation going.
Hello everyone, my name is Elliot Cornett, and I am more than excited to talk to you about my senior research project, which was a low-cost approach to reverse osmosis desalination using solar energy. And I realize that might sound a little confusing right now, but I promise by the end of this presentation, you'll understand a little bit more. So just for some quick background, right now, one of the biggest problems facing our planet is the lack of clean, accessible drinking water. So almost 800 million people worldwide lack the access to consistent, clean drinking water, and around 4 billion people experience severe water scarcity for at least 1 month out of the year. Now these are some really alarming statistics, and what are we doing to sort of combat this? Well, one of the biggest things is a process called saltwater desalination, which is essentially the process of taking salt water and applying pressure to it through a special kind of membrane that turns that salt water into a form of water that's drinkable for humans. And especially with the effects of global climate change really reducing the amount of freshwater resources available to us, saltwater desalination has been more important than ever. Right now, this technology is really only used in places like North Africa and the Middle East, where they are experiencing severe droughts and severe losses in their freshwater resources. However, as this technology expands, it will definitely be applicable in different areas of the world. So this is Ashkelon Desalination Plant. It's in Ashkelon, Israel. It's the largest desalination plant in the world, and it produces around 330,000 cubic meters of water per day. Now that's a lot of water. However, one of the biggest problems with this is that most of that water isn't actually going to people who need to drink it. Most of that water is actually going to oil companies and other large corporations that are using the fresh water for other things. Another thing is that a plant like this, in order to generate enough energy to sustain a plant like this, they're doing a lot of things like burning fossil fuels, particularly oil, which is not ecologically sustainable at all and is really bad for our environment. Which is why the purpose of my project was to be able to utilize green energy in a sustainable way that was also way more affordable than the current method and able to be used at the point of contact, meaning that anyone who had access to this technology would be able to use it wherever they had salt water. So what is reverse osmosis? I'm so glad you asked. It all starts with the feed solution, which is the salt water or ocean water, and it enters into one end of the membrane. As it enters into the membrane, it'll encounter several layers, the first of which being a feed channel spacer. Now this is just a sort of netting that allows the water to actually have space to move within the membrane, as it is pretty tightly wound up. And then it will encounter the actual membrane itself, which is a semi-permeable plastic that allows water molecules to pass through but keeps salt on the outside. So as pressure builds up in the vessel, the water will end up being pushed through the membrane and end up in a permeate collection material, which is similar to the feed channel spacer in that it just provides space for the water to move around. And all of this is held together with an outer wrap, which is just a structural component that keeps things nice and tight. So as the permeate collection ends up spiraling towards the center, it'll collect in the permeate collection tube and end up as permeate out one end of the vessel. And then all of the remaining water that didn't go through the membrane will end up as concentrate. So how did I go about designing my membrane? Well, it was a pretty simple design philosophy. What I would do is I would examine the materials that were used in industrial systems, and I would try to find low-cost alternatives to those materials. So for example, a lot of industrial systems will use a material called Vexar netting, which is this super expensive netting material that essentially does the exact same thing as a screen door. So I took a bunch of screen door material, which is way less expensive, and used that instead. And then I repeated this process for all of the materials that I use in my design. As you can see here, the outer wrap is duct tape, and I'm using PVC pipe and brass fittings and other relatively low-cost items. My project also contains a 25-watt solar panel, as well as a 12-volt 35-amp-hour battery for that solar energy aspect that I mentioned earlier. So this is a solar panel system. As you can see, the solar panel is right here. It is charging a battery, which is off-screen, and then that battery is plugged into a controller, which allows me to turn on and off this pump, which generates pressure for the design to work. So this is what the design looks like in action. As you can see, the fully charged battery right here is plugged into the controller, and if I can get my labels to come up. Yeah, this pump is pumping this salt water, the feed solution, into this membrane, and the membrane is then allowing the concentrate to collect in the yellow bucket, whereas the permeate will go all the way through this vinyl tubing and into the white bucket. So how much did this cost? I'll try to get through this pretty quickly because I have limited time up here, but essentially the biggest thing is that the biggest, most expensive part of the RO membrane itself is going to be the diaphragm pump that I use, which costs around $40, which accounts for about half of the cost of the membrane itself. However, the most expensive part of this project by far was the solar panel system, which in total cost around $120, bringing the total to $207.94, which sounds like a lot, but it's actually a lot less than a lot of home systems that you would find on the market today, which generally cost around $300 to $500. Especially considering this is you're able to use this in any area of the world as long as you have access to the sun. It's really not a bad cost. But if you wanted to implement this into a place that already had electricity and you didn't necessarily need the solar panels, then this cost would be brought down to around $85.45. So how did I test my design and see if it worked or not? Well, the first thing I did was a freshwater test. So I would just send fresh water through just to see how much would actually end up permeating through the membrane. And then after that, I mixed a special solution of salt water, which was around 35 parts per thousand. And then I tested the salinity of that water before and after using a salinity refractometer, which I don't really have time to go into, but ask me about it later because it's really cool. You're all looking at me, I know what you're thinking, did it work? And the answer is no. Specifically, my experimental value for the permeation was around 2.86, where that should be around 25%. And for the desalination rate, the experimental value was around 9.38%, whereas ideally this would be around 95%. Now, these statistics are obviously not up to par with what you might expect from a commercial reverse osmosis system. However, I do have some really good ideas on how I would improve this in the future. And I was asked, like towards the end of my research, I was basically begging my mentor to allow me to have like a couple more weeks because I was so close, but I digress. As for future work, one of the biggest issues that I ran into was that the vessel was not actually being pressurized enough. So obviously for this system to work, there needs to be a lot of pressure, as that's one of the biggest problems is generating enough energy to create that pressure. But the problem with my vessel is because of the materials that I used and the way that I assembled them, a lot of the water was just finding the easiest way to exit the vessel, which happened to be just going around the membrane instead of going into it. So I would definitely want to include a pressurized valve system to the end and hopefully generate more pressure for the vessel to actually work. Also, I want to experiment with other membrane materials because I was not able to access a lot of materials that are available only to corporations, and I was only able to access like consumer materials. So like for example, my PVDF membrane, which is the plastic that I use, costed around $15, where there are other materials that cost a lot less that I was not able to access. I would like to acknowledge Campbell Hall and the University of Alabama at Birmingham for allowing me to use their labs for the duration of my project, as well as my wonderful mentor, Leslie Hendon, who helped me through every step of the way and was there to give me suggestions and point out the things that were wrong with my design and help me in every way. I would also like to thank ASFA for providing some of the materials that I used during my project. Thank you so much, and I hope you enjoy the rest of the project.
Hello everyone, my name is Henna Craig and I did my research at UAB, and I did my research, it is a bioinformatics study of the TET2 ASXL1 microenvironment. So myelodysplastic syndrome, also known as MDS, also known as pre-leukemia, is any disorder in which white blood cells do not form or function properly. So the overall goal of my experiment is basically to find genes that are overexpressed in MDS so that we can potentially find drugs that inhibit the overexpressions, and then we would prevent people from getting AML, which is acute myeloid leukemia, which is the next stage progression of this disease. So there are many different mutations that can occur in someone that has MDS. The ones that I'm looking at are TET2 and ASXL1. TET2 and ASXL1 deal with epigenetic regulation, also known as the formation of DNA. So when you have a mutation in genes that cause the formation of DNA, you get mutated DNA, which is how we get cancer, which is how we get leukemia. So I had two objectives to my project. My first objective was to find different gene networks in a mouse that with the TET2 ASXL1 mutation, and to also classify all the cells in the mouse using the whole bone marrow microenvironment. My second objective was to treat stem cells and determine which genes were increased at 1 hour or 6 hour intervals. The stem cells were also TET2 ASXL1 stem cells. So a little bit of background. The cytokines that I used for my treatment were IFNG, IL-1, IL-6, and TNFA. All of those cytokines are already upregulated in MDS. So basically the purpose of the treatment was to see, is this cytokine causing something else to happen later down the line, and is that the root cause of MDS? We also use the serum as a treatment. The serum is basically just the outer layer of the bone marrow, has a lot of inflammatory properties in it, so we also use it as a treatment. This is just a little diagram of what the treatment looks like. PTN is a protein-coding gene. This is a chronic myeloid leukemia stem cell. So when there's a lot of PTN, JUN and UPR are increased. Those are both two genes. And then if we add anti-PTN or like an inhibitory drug, then JUN and UPR decrease and apoptosis increases, which is exactly what we want. So prior to my work, single-cell RNA sequencing was done on a mouse model, both wild-type and TET2 ASXL1, because all of my data is pooled. And then there was cDNA, and all that cDNA was sequenced into nucleotide barcodes, which then I uploaded into Partech, which is the bioinformatics software that I use. So I did all the exploratory analysis for this project. So after I uploaded everything, I had to filter it. So basically what this does is it excludes any genes or any like cells I might have gotten into the cDNA when the graduate students were doing the single-cell RNA sequencing. So that includes any like red blood cells that might have gotten into the mix. We also filter out anything that has a mitochondrial rate of 10% or above. That means that the mitochondria is damaged. So we do that because we don't want the cell to become a different type of cell. Then I did a normalization on the data. So normalization takes all of my data into a log2 transformation, and this actually lets me do statistical analysis on the data. I also put a noise reduction filter on it. The noise reduction filter just filters out any genes that only occur in the data set 0.01% of the time. Then I had to reduce the dimensionality. This is my favorite part because you get a very nice little visual model at the end. So first, I had to do a PCA, which is a principal component analysis, and all that does is it takes the log transformation and puts it onto a 3D graph. Then I had to do k-means clustering. So k-means clustering uses differential analysis to try to group different genes together based on what they have in similar to each other. So I had 25 different clusters, or you could call them 25 different cell types. And then I did a t-SNE. But before that, I had to classify all of my cells. So for the whole bone marrow, I did a gene-specific analysis. Basically, that just takes the k-means clustering, and it says cluster 1 versus all the other clusters. What genes are in cluster 1 that are not in the other clusters? Copy and paste that list, and then put it into a site that's called Enricher, and Enricher would give me back a cell type. For the HSC data set, or the treated data set, all I had to do was the gene-specific analysis because we already knew that we wanted to do the cells in a treatment type and how long the treatment was done for. So these are my t-SNEs. They're very colorful. So figure 9 is the whole bone marrow with the classifications. On the left, you can see all of the cell classifications. And then figure 10 is the HSC data set with all of the cytokines and their treatment types. Okay, so after that, I had to do pathway analysis. So this is the pathway enrichment of macrophages. It's just an example. We look at enrichment score and p-value to determine which pathways we want to potentially look at to see if they have any genes that are druggable. Again, this is an exploratory analysis, so you can essentially look at whatever you want. I chose to look at osteoclast differentiation, has a very low p-value and a high enrichment score. So the osteoclast differentiation KEEG for macrophages looks like this. Anything in red is overexpressed. Anything in green is underexpressed. These are all genes. So I specifically looked at the overexpressed genes because the end goal of this project is to find drugs that will potentially decrease the overexpression. Specifically, I looked at genes that looked at cell transcription, cytokine interactions, or like immune cell signaling and cell proliferation. So the two most interesting ones were like JAK and STAT. They form the JAK-STAT signaling pathway, which I will show in the next slide. And then also AP1, which is another gene that aids in transcription of DNA. So this is the JAK-STAT signaling pathway. So we have JAK and a cytokine, and then we have the STAT that comes in and recruits the JAK-mediated phosphorylation, and then it ends up actually transcribing DNA. So if we have more, JAK and STAT, then that means that we have more DNA, which increases the likelihood that it can be mutated, which is where we get cancer from. So these are some of my results. Again, this is exploratory. You can look at whatever you want. I chose to look at IFNG MDS 1 hour showed JUN. IO1 was MDS 1 hour showed IL-6, which actually deals with B cell signaling and inflammation. Sera 1 hour is SRF, which deals with cell proliferation and differentiation. And then the macrophages was the transcriptions. CD4 T cells had GRAP2, ROR alpha, and JUNK. And then neutrophils had CD38. So for future experimentation, I did try to grow stem cells, okay? But it's very hard to grow cells that grow other cells. I tried 3 times, and every single time they died. But I'm currently in the process of growing them again. I actually have to go tomorrow and change out media of the cells. So I'm going to try to grow those again and actually use drugs to inhibit the overexpressions of some of the genes that I found. And I would like to acknowledge the following people: Dr. Robert Wellner of the UAB Comprehensive Cancer Center. He was my mentor. I would like to thank Ms. Thrash for helping me with all of the research presentations and putting together this PowerPoint, Mr. Ware for being an amazing faculty advisor, and Ms. Chin for everything that she's done for the math science department, and of course, my family. And here is my references. Thank you.
All right. Hello, everyone. My name is James Downs, and I'm a senior here in the Math Science Department at ASFA. And I did my senior research project on the relationships between corneal resistance factor, corneal hysteresis, and ocular rigidity. So that's a lot of big words, so I'm going to explain some of those. And so first, intraocular pressure is the pressure on the inside of the eye, and ocular rigidity is associated with the basically changes of pressure inside your eye, and these could be from anything from a change in volume, from fluid in your eye, or just rubbing your eyes, or blinking, or anything that basically changes the shape or the volume of your eye. So if you have a higher ocular rigidity, basically when these changes happen, the eye wall is less likely to be able to stretch out and absorb that. And so that just causes a pressure spike, and we think these transient intraocular pressure fluctuations, that they are very important in the onset and progression of glaucoma, which is one of the leading causes of irreversible blindness in the Western world. The problem with getting ocular rigidity data is that it can only be measured invasively. So the goal of this project was to find an alternative by using a clinical device in order to be able to predict the ocular rigidity so that we can see if someone's at risk for glaucoma and then start treatment. So we tested the hypothesis that corneal resistance factor and corneal hysteresis are biomarkers of ocular rigidity. So as I said, the only way to get the ocular rigidity data is invasively. So we used a colony of rhesus macaque monkeys, which look like that, at UAB, and they are instrumented with wireless radio telemetry systems that measure the intraocular pressure 200 times a second, 24 hours a day. And how we got the data for corneal hysteresis and corneal resistance factor was with the Reichert Ocular Response Analyzer, which is, it looks like that. If you've ever been to the eye doctor, you've probably had it done to you. So it's just a clinical device that uses an air puff on your eye and then uses a laser to measure how much your eye actually deforms with that air puff. So basically, the parameters from that are corneal resistance factor and corneal hysteresis. Corneal resistance factor is the measure of the elasticity of the cornea, and corneal hysteresis is shows how much energy was absorbed by the eye during the air puff. And we used 9 male NHPs, which is non-human primate, and we used all normal eyes in the experiment because some of them were actually developing glaucoma, so we didn't want to use those eyes and have it affect our data. And then with all that data, we were able to plot a regression and to see if they're associated. And so from that, we got this graph matrix, which is a matrix of the scatter plots. And how these work are, what is up on the top, that is what's the Y value that's plotted on the scatter plot, and then what's off to the side is what is actually plotted on the x-axis. So this one right here would be ocular rigidity and corneal hysteresis. And from this, we found that corneal resistance factor and corneal hysteresis are both significantly positively associated with ocular rigidity, but due to the small r² value, the r² value means that if it's only 0.1, that means that only 10% of the variance of the ocular rigidity was accounted for by these other, by corneal resistance factor. So as you can see, it's not a very strong line like this one. These two were already found to be, these were already known to be very strongly correlated. So we were looking for something sort of like that, but we didn't really get that. So if you were given one of these corneal hysteresis or corneal resistance factor values, you wouldn't be able to really predict the ocular rigidity. So that's where we come to the conclusion. We used IOP magnitude, which is just the change in intraocular pressure during the air puff. That was just to double check because those were already known to be associated, just to make sure we were doing everything correctly and our methods were all sound. And while corneal hysteresis and corneal resistance factor are significantly positively associated with ocular rigidity, the predictive utility of them is very limited because of the low r² value and the small effect size. The effect size is basically the slope of that best fit line through the scatter plot. And with that, given that scatter plot, you wouldn't be able to predict ocular rigidity very accurately. But our p-values were very low, and the relationship was very significant, but it just wasn't strong enough, and the predictive value was not enough to be able to actually be used in just at the doctor's office. So the same thing was found with IOP magnitude, and so what we plan to do in the future is to use a different device because it wasn't anything that we did wrong during experimentation, but we plan to use a different device called a Pascal DCT, which provides a different variable called ocular pulse amplitude, which is just the change in pressure from just the influx of fluid into the eye, because it's kind of like a contact that goes on your eye, and then with that it can only measure the ones from the fluid and not from blinking or moving your eye, because it's actually physically on your eye and you can't close your eyes. So we're going to see if that works out instead. So I'd like to thank my mentor, Dr. Downs at UAB, and his whole lab for being very helpful. And I'd like to thank Dr. Legrone for being my faculty advisor, and Ms. Chin for just everything. So yeah, and there are my sources. Thank you very much.
Hello everyone, my name is Trisha and I'm a senior here at ASFA, and my project is on exploring the local and global factors of the generalized Polygraph illusions. So before I begin, I want to ask a question, and that is, what is an illusion? And so I'm sure we've all seen illusions. They're a great way to entertain people, to kill some time, but they actually have a second and more scientific purpose, and that is to understand the limits and capacity of our perceptual apparatus. And moreover, they can specify how the constraints of perception are set. Moreover, they let us analyze the cognitive sub-processes underlying our perception. And I plan to do this by looking at the Polygraph illusion. So the Polygraph illusion can be described as a visual illusion in which two line segments are terminated by two parallel lines, in which these two line segments appear to be non-collinear or misaligned, when in fact, they're the same line. They're completely collinear, completely aligned. So why do we see the Polygraph effect? Well, researchers postulate that it has to do something with the single end-stop neuron in our visual cortex. So a single end-stop neuron is a feature detector, and what feature detectors do is it focuses on some part of an object or a line. And this specific feature detector, a single end-stop neuron, it focuses on the length of a line. So here I have a representation of how it works. I have different lengths of a yellow line against a black bar. Now when the length of the yellow line is the same length as the black bar, we get the most amount of cell response. However, if it's too short or if it's too long, we get the least amount of cell response. And so researchers think that the line shown in the Polygraph illusion is too long, our brain cannot process it, it has very low cell response, and therefore we see the effect the way we do. Now, previous work using the Polygraph illusion used binocular disparity, and it made the oblique line, that means the two line segments, near, far, or in the same plane as the blocker. The result was it's much less common to see misalignment in the near condition, which can lead to a conclusion that the interaction between the classical receptive field and the inhibitory end zones is asymmetric and binocular disparity dependent. And the key thing about this study was that the depth order was implied directly and explicitly. So in my project, I wanted to imply the depth order using shadows, which is an indirect method of manipulating depth order. And so I hypothesized that when the oblique line is in front of the block, the junction detector will not be activated, and the distortion of the oblique will not occur. And second, when the oblique line is behind the block, the junction detector will be active and a distortion will occur. So to begin methodology, a participant would sit in front of this 20-inch Sony CRT monitor. They will be looking at different conditioned stimuli, and they will place their head in this headrest in which the chin and forehead placements are adjustable so that their eyes are level with the center of the screen. Next, the software that I used to create the conditions and the stimuli was developed in a laboratory that uses OpenInventor and OpenGL graphics libraries. Moreover, the participant would have to sit 57 centimeters away from the screen. And this is because when we look at an image, that image is displayed on our retina, a lot smaller version of it. So at 57 centimeters, 1 centimeter on the screen is going to equal 1 degree of visual angle. So when we're looking at illusion, we can calculate the misalignment that we see, convert that into degrees of visual angle, and then convert that into arc minutes to actually find how strong that illusion might be. So these are the conditions or the stimuli that I created. The first two are just the generalized Polygraph illusion, a left and a right version. Conditions 3 and 4 are what I call the naked Polygraph, and I kind of use them as a control. So you can probably already tell, but the misalignment is greatly reduced. And that is because previous studies show that this junction that the rectangle and the line creates is removed. And so we don't see the illusion the way we do. Now, conditions 5, 6, 7, and 8 are where I'm actually doing the manipulation of depth order using implied shadows. So for conditions 5 and 6, the shadows imply that the triangle is in front. And for conditions 7 and 8, the triangles imply that the shadow implies that the triangle is behind. And so when a participant is looking at these conditions, or these stimuli, they can actually adjust a part of it, and it's called a method of alignment. So for example, if they were given this stimuli right here, they can actually use the up-down arrow keys to adjust the bottom part of the stimuli. So for example, they can move it up, up, or down until they think that the two lines are completely aligned. If they were given a stimuli or a variation like this, they can actually use the left or right arrow keys to move the bottom part of the triangle out. So until they think that hypotenuse is completely aligned. And now, so a participant will sit down to begin the simulation, press the space bar. Then they will use that method of alignment that I described. Once they're completely satisfied with the alignment, they press the escape button. And then the monitor will actually record the number of key presses that the participant moves. And I had 8 conditions. I did a total of 10 trials. So that leaves to the participant doing this iteration a total of 80 times. And so, unfortunately, my research report is still being reviewed by the IRB, and so I only have preliminary data from my mentor, ACD, and from myself, TB. So what I did is I took the data that the monitor recorded, those key presses, and I imported into a MATLAB function that was able to create these bar graphs. So what is shown in these bar graphs is each condition and its corresponding illusion bias, or how strong the illusion is. And this was calculated using that 57 centimeter ratio. Moreover, I subtracted conditions 3 and 4 from all of the conditions because everyone sees things differently. We all have a different perceptual set. So if we're able to subtract these controls, we can get a more accurate representation of that human's perceptual system. And so this is my mentor's, this is mine. And like I said, these are preliminary data, so I can't really refute or verify a lot of the conclusions I made. But there were a couple of interesting things. The first thing being that if we look at conditions 1 and 2, this is my mentor's. Conditions 1 and 2 are just the same illusion, just flipped, but for some reason there is a left-right asymmetry. For example, condition 2 has a really high illusion bias when it's just the same thing. And there are a couple of things that I hypothesized that might be happening here. First, it could be age. It could be left or right eye dominance, which could be an interesting data point to collect in the future. But I also hypothesize this discrepancy can actually be gone in the future once we get a lot of participants. Moreover, these preliminary results show that the Pottendorf illusion was visible in all the conditions, and so I can move on knowing having that confidence. And this leads me to my future work, of course, being getting more participants. That would be 18 plus students at UAB. The participants will follow the same methodology and the same data analysis would be done. I would like to give a huge thank you to my mentor, Dr. Alan C. Dobbins, Mr. Sproul, my faculty advisor, Ms. Thrash and Ms. Chin for helping me put together this presentation, and of course my family for supporting me. Here are my sources, and thank you for listening.
Hello everyone, my name is Maya Lundy and I did my research project on analyzing organismal size among life cycle stages in Gracilaria vermiculophylla. So Gracilaria vermiculophylla is a type of macroalgae, sort of like a seaweed, and it's a widespread invader that is affecting marine ecosystems throughout the northern hemisphere. We know that organisms that sexually reproduce can alternate between haploid and diploid nuclear phases. So a diploid organism can produce haploid gametes that later on will recombine to produce a new diploid organism. However, for some organisms, such as the macroalgae that I studied, either stage can undergo somatic development. So a haploid gamete can either recombine with another gamete, like I said, or it can experience development of its somatic cells, which are like a human's body cells, and it can exist as its own independent organism. And these independent gametes are what we call tetrasporophytes. But according to all genetic models, this sort of life cycle should not be able to persist. And we even see that during the invasion of soft sediment habitats, the gametophyte stage is consistently lost. So that poses the question of how is this life cycle able to persist? And knowing this, I wanted to figure out if there are observable and measurable phenotypic differences between haploid and diploid stages in this macroalgae on the eastern shore of Virginia. And the goal of this research project is to describe these differences using observation. And if the differences are significant enough, then this would suggest that organism size has an impact on structural and physiological aspects of the algae. Previous work has hinted that slight differences among stages can lead to the evolution and maintenance of haploid-diploid life cycles. And we even see that in the non-native thalli, tetrasporophytes were larger, and we know that it's advantageous for invasive species to be larger than the native species in order to outcompete them for resources. And we see an example of this in free-floating populations. Tetrasporophytes had a lower protein content and required more tensile strength in order to pull a branch off the main axis. And this could possibly hint at the reason why this stage is dominant in free-floating populations. So in order to do my research, I received samples from 3 fixed sites and 2 free-floating sites. And a fixed site is where the thallus is attached to a hard substratum, such as a rock or in the ground. And a free-floating site is where the thallus is not attached to anything. And in the 3 fixed sites, all 3 life cycle stages were represented, male, female, and tetrasporophyte. And in the free-floating sites, they were dominated by the tetrasporophyte stage. So then each sample was photographed individually in a Pyrex baking dish set on top of a light box, and a ruler was included for calibration of the imaging software ImageJ. And then I used ImageJ to measure the main axis length, thallus area, thallus perimeter, and to count apices, which are like the tips of the ends. And then I put all this data into a spreadsheet, and then natural log transformed the data in order to normalize it, and then ran an ANOVA test using R. And then I measured the means and standard deviations of each variable, and then graphed them with error bars, so 2 standard errors, and I have 3 graphs here today. So post-hoc test results and graphical analysis show that for the female tetrasporophyte stage, they were about the same axis length, but males were for sure shorter. And then area, the tetrasporophytes dominated, and the males had the smaller area. But what was interesting to see was males actually had the largest tipper area, which means that they were bushier. And this could be because they stopped thallus elongation in order to produce more gametes. But overall, in the tetrasporophyte stage, thallus size was larger, which could contribute to their dominance while free-floating. But this could also be because in the 2 free-floating sites that were sampled, tetrasporophytes were the dominant stage anyway. But this could also contribute to the idea that the tetrasporophyte stage has a significant enough ecological advantage to develop as a new stage. But in conclusion, differences in morphology provide new hypotheses to test about how organismal size can influence the success of different stages and ultimately lead to the maintenance of morphologically very similar stages within the same life cycle. I would like to thank my mentor, Dr. Stacy Kruger-Hadfield, and my postdoc, Dr. Will Ryan, and my faculty advisor, Ms. Thrash, and the math science chair, Ms. Chin, for everything she does. And then I'd like to thank everybody in the Kruger-Hadfield lab and everybody that provided the funding for this project. And thank you guys for listening to my presentation.
Alright, hello everyone. My name is Pranav Janasekar, and tonight I'll be talking about exploring the use of PCR to identify a population of mice deficient in Rna 1. So for some background, cardiovascular disease is the world's #1 killer. It kills about 18.6 million people a year. And there are many different types of cardiovascular disease, but one of the most common ones is coronary artery disease, which is what I'll be talking about today. And coronary artery disease is basically when plaque builds up alongside the walls of the blood vessels, and it makes the arteries smaller, and it makes blood flow to the heart, or it restricts blood flow to the heart. And there's been many genes that have been linked to cardiovascular disease present in the walls of the blood vessels. So what is LMOD1? LMOD1, or also known as LMOD1, is a protein encoding gene, and it's present across many different species, including humans, mice, and zebrafish. And it's mainly expressed in smooth muscle tissues, such as the aorta, the stomach, or the bladder. And what we know about smooth muscle cells is that they're critical to the structure of blood vessels themselves. And a previous study has shown that mice deficient in LMOD1 die soon after birth because they have a defect in peristalsis. This is because the smooth muscle cells in their intestines aren't able to contract, so because of that, they can't digest food properly. So my main objective was to find a select population of LMOD1 deficient mice and use an existing breeding plan created by my lab and PCR methodology. So if you look at the breeding plan that we have over here, there's two different scenarios. Either a LMOD1 wild type and a heterozygous mice can be mated together, or two heterozygous mice. And both of these scenarios have a chance of producing offspring that are also heterozygous. Now the reason why the heterozygous mice are important is because my lab is using a mouse model that only uses heterozygous mice in order to find out more information about the role of LMOD1. And then in order to genotype the offspring from the 8 mice that I was given, I had to use PCR methodology. So using all this, I created my hypothesis, which was that using the Mendelian ratio, the predicted genotype would be in a 1:2:1 ratio, which is the ratio between knockout, heterozygous, and wild-type mice. So for my methods, the first thing I did was isolate and purify the DNA from the mice. So I started off by collecting my mice tissue, which was about a 1 centimeter long tail snip of each of the mice. So once I had the mice tissue, I had to purify and isolate the DNA from it. So I used a commercially available tissue mini prepping system in order to obtain my purified samples of genomic DNA. So once I had those, we wanted to get the DNA specific to LMOD1 itself. So I combined those samples with specific primers that targeted a specific portion of exon 1 of LMOD1. And then after that, in order to amplify the amount of DNA we had to make it easier to visualize, I used a PCR, which will heat and cool the samples over and over again in order to make the DNA replicate. So to analyze the data, I started off with agarose gel electrophoresis. So if you see over here, there are these sample wells over here, which is where I put in my DNA samples, and then I ran a current through the gel. What the current does is it makes the negatively charged DNA get attracted to the positive side of the gel. And because of the structure of the gel, the smaller fragments of DNA travel at a faster rate, so it ends up creating bands like this, where the shorter fragments of DNA travel further along. So once I finished running the gel, I took it to an imaging machine, which produces an image similar to this, because the imaging machine has a UV light that shines through the gel, and the DNA samples contain a dye that reflects UV light. So that's why you can see all of the bands here. So after that, I analyzed the agarose gel, and that's how I was able to determine the mouse genotypes. So this is my result. This is a completed gel that I took a picture of in the imaging machine. So what you see here in the first column is the molecular weight ladder, and the reason why you see the 414 and 256 base pairs marked is because that's the range of exon 1 that we targeted. And then the first column itself is a molecular weight ladder, so that's pretty much like a ruler telling you how big the fragments of DNA are. And in the second column, we have the control group. So this is DNA from a mouse that we already know is heterozygous. So then we can compare the next 8 samples, which are the ones that the samples that I made, and you can compare those to the control group, and you can tell that 8 of them are heterozygous because they're almost identical to the control group, and then the one single band represents a wild-type mouse. So my conclusion was that out of a group of 8 mice, I was able to find 7 heterozygous mice and 1 wild-type mouse. So the actual ratio ended up being 0 to 7 to 1, instead of the 1 to 2 to 1 like I predicted. Now the main reason for this is because I didn't know that The knockout mice, they die soon after birth. I didn't know about that until after I made my hypothesis. And all of the mouse tails that I obtained were from mice that were several weeks or to a month old. So there's no way I could have gotten any knockout mice because they would have been dead already. But I was still able to satisfy my main objective, which was to reveal the use of PCR to identify a population of LMOD1 deficient mice. So for future studies, I plan on validating these results using alternative technologies such as Qpcr, which will give me higher resolution data, and Western blotting, which is similar to the agarose gel that I ran, except instead of separating the DNA into fragments, it separates proteins. And I'd also combine that with a larger cohort of mice instead of 8. 8 is all I had available at the time, but next time I'll try something like 20 or 30 mice in order to get more accurate data. And what I can do with these heterozygous mice is build up the population in order to continue using the existing mouse model in the lab and further investigate how LMOD1 provides support to blood vessels. And I'd like to say thanks to my family, my mentor, Dr. Nanda, my advisor, Mr. Kwong, and Ms. Chin.
Hello, my name is Lizzie Lancaster, and my project is about understanding the concentrations of particulate matter across several cities in the state of Alabama and how it's related to the people that live there. So particulate matter is an air pollutant consisting of solid particles and liquid droplets suspended in the air. There's two main categories of particulate matter. There's PM10 and PM2.5. PM2.5 is the smaller of the two. It's about 2.5 micrometers in diameter. Looking at this strand of hair, we see that the human hair is about the size of 50 to 70 micrometers in diameter, and PM2.5 is about a 20th of the size, so it's extremely small, which is related to why it has a negative impact on human, plant, and animal health, as well as damaging buildings with prolonged exposure. Particulate matter is a contributor to reduced visibility in the United States, and Alabama currently has the 12th worst air quality in the US, which is why it's important to understand how it's related to Alabama. Particulate matter can come from sources like natural sources like pollen, or single sources like dust from unpaved roads, construction sites, fires, but most of it, the reasons why it's considered an air pollutant is the high concentrations from the chemical reactions of sulfur dioxide and nitrogen oxides from automobiles and power plant exhausts. So like I said before, its impact on human health is that with its size, it's able to penetrate the lungs and reach the bloodstream, and is linked to issues like aggravated asthma, decreased lung function, irregular heartbeat, non-fatal heart attacks, and overall increased respiratory and cardiovascular issues, which is especially vulnerable, or especially an issue for those that are considered vulnerable, so those under age 5 and 65 and older, because their immune systems are generally less functioning as well as the rest of the population. So the objectives of my project were to look at the concentrations of PM2.5 across 9 cities in the state of Alabama in the year 2019, compare these averages among the cities, as well as make predictions for the rest of the state, and understand how it's related to the people currently living there. So to do this, my data was collected by the Alabama Department of Environmental Management, ADM, in the year 2019, and I determined the averages of each of the 9 cities. Here we see is the example of a possible air quality monitor, which collects particulate matter samples. For my population data, I used 2019 estimates based on the 2010 census to determine the total population as well as the at-risk population. Like I said before, those under age 5 and 65 and older. So a large part of my project was actually my ability to learn how to use the new software. I'm not personally a computer science person, so this was a big feat for me to be able to do this. So ArcMap is a child of Esri's ArcGIS. It's a geospatial processing system to understand, analyze, and view real-world data, which I use to understand how particulate matter is spread across the state.
To do this, I compiled and graphed the concentration averages according to their geographic location and interpolated the data using a method called the kriging method. Then, testing for correlation to understand how the people are related to the particulate matter, I looked at the 2019 average concentrations and population density, as well as the average concentration and percentage of average populations. So here we can see is my interpolation map that I created in ArcMap. Here we see Tuscaloosa has the highest concentration of particulate matter, and it looks alarming just because it's in red or like a bright pink, but it is under the EPA's annual standard of 12 micrograms per cubic meter for an entire year, so the average. So they're all at a healthy level. We just see that Tuscaloosa has a little bit more particulate matter than other areas of the state. Now looking at the comparison of the cities among all of each other, we see across the board with the 2 standard error, which is a 95% confidence interval, that there's overlap between the bars, meaning no site has significantly more or less particulate matter than any other city. However, if we look specifically at Tuscaloosa, Decatur, and Crossville, we see Tuscaloosa has a little bit more particulate matter than Decatur and Crossville, which is what we see in the map as well. And the next part was looking at the people and the particulate matter. So looking at the population density with the average concentration, we first see a positive trend, and I use Pearson's correlation test and the p-value test to test the significance. We see that there is a correlation, and this is significant, meaning when there's more people in these cities, there's generally more particulate matter. What surprised me personally was the relationship between the at-risk populations and average concentration. We see a negative sloping trend where there's generally less particulate matter when there's more vulnerable people, according to like percentages. And Pearson's correlation says there's a relationship, however, the p-value test, so there's a 14% chance that this is actually due to chance and not a relationship. So I'd like to conclude, like I said before, there's a strong positive correlation between average concentration and the population density. And while there's only a trend with the percentage of at-risk populations in the average concentration, it's a good thing that there's a negative sloping trend, but it's always good to help lower it. So in the future, what I'd like to do personally, as you can see, we don't have data from Huntsville and Birmingham, which are kind of the metropolises of the state. They publish their own reports at the end of every year, so the state of Alabama does not have access to their data, so I was not able to access their data. But I also like to increase the sites collecting data here in the northwest region and the southwest region of the state to make better predictions according to the Kriging model. I'd also like to look at a change in the presence of time. PM 2.5 can differ across the times of the year. So say in the spring, currently, we have a lot of things blooming, so there's lots of leaves to collect particulate matter on its leaves and help reduce that concentration of particulate matter. However, we also have pollen being produced, which kind of affects everybody's allergies, but also increases the amount of particulate matter we see. So it'd also be interesting to see about the time of years, so like at different times, like different years per se, so like 2015 versus now, and even 2020 when we had COVID and kind of changed how we run our society to make better predictions for the future and how we should go about changing our policies. I'd also like to look at different relationships among other variables. What I'm currently looking into is how it's related to vegetation distribution. Here we see Landsat imaging, or a satellite image of one of the cities that I was looking into, Crossville. We see it's an NTVI distribution, so it's a natural distribution of vegetation index. We see the red is where there's no vegetation or very little, and green is where there's high concentrations of vegetation. What I'm currently dealing with right now is cloud correction. Here we see in the areas that have like a little bit of muddled green or like a little bit of orange, that's where a cloud is, and is kind of not giving us the accurate representation of how much vegetation is in these areas. So using this, I want to see how it's related to the people there, as well as the particulate matter, to see if it's useful to suggest increasing your vegetation in these areas to help reduce the particulate matter and help the health of these citizens. I'd also then like to look at sources of pollution, how it's laid out across the cities, to see if it'd be useful to make more regulations about having pollution being produced, as well as helping the health of these people living there. And I would like to thank my mentor, Dr. Miriam Karimi from UAB, as well as my faculty advisor, Ms. Thrash, and the rest of the ASFA faculty and my family for help throughout this project. Thank you. my project is on verifying FGH-HPRT and proving the Chang-Feder-Grunbaum theorem is induction. So imagine placing an ant on an empty surface with no obstacles. The walk that it traces as it wanders away from the origin could be considered as a random walk. But my project is focused on two types of random walk. The first type is a simple random walk, and it is a walk determined by up and down steps of equal probability at 50%, while we're more focused on another type of random walk, which are random walks generated by random permutation. And these walks are determined by the orders of numbers within a permutation. So if two adjacent numbers are increasing, then that's an up step. And if similarly, if it's decreasing, that's a down step. So why is it important? It is important because it can be used in numerous areas of biology, such as genetics and bacterial migrations, and it can also be used in computer science as a way to sample large-scale peer-to-peer networks. And other studies have actually shown that random walk models offers the most convenient and accurate solution compared to other stationary models, such as random correlation or normalization. So for my project, my first goal was to prove, verify, or disprove the FGH-HPRT conjecture. And it is a conjecture by these people with a formula shown here. But in other words, it means that a random walk generated by random permutation has the same midpoint distribution as that of a simple random walk. And for the second part of my project, it is to prove the Chang-Feder-Grunbaum theorem is an induction, and the formula for that theorem is shown here, and it's simply the midpoint distribution formula for a simple random walk. So for my project, I have first written two computer programs using MATLAB and Java to mimic the random walk generated by random permutation. And then I collected results up to n equals 100 and verified these data against the expected results from the conjecture. And I searched through to see if there were any more patterns of interest that could help me prove the conjecture. And then I moved on to a slightly different topic, which is to prove the Chang-Feder-Grunbaum theorem is induction. And lastly, I further explored and see if possible if there are any more side results that can help us prove the initial conjecture. So here is a simplified computer algorithm that I wrote for the project. So this part is entirely for generating an array of all unique permutation and can have. So what I did is I generated an empty array to store this permutation with a size of n factorial. Then I generated a new permutation using the permutation class as shown here to generate a new permutation, and then I check if the new permutation is already in the array. If it is already in the array, simply generate a new one until I haven't reached one that has to be stored in arrays. And then I check if the permutation array is full. If it's not full, I simply repeat the process over and over again until I reach one, until the entire array is full, and at that point, that means all the possible n permutation has been generated. So the next two steps are a lot simpler, which is to use the permutation arrays that we just generated to generate all the walks, and then calculate the number of positive steps for each walk, and lastly, to categorize the number of positive walks with the number of positive steps. So for example, here are two of the sample results that we have. One is for n equals 7 and one is for n equals 8. For example, when n equals 8, that means there are 11,025 n equals 8 walks that have 0 positive steps. And here is a simplified proof by induction. So for the alpha function, we have this formula shown over here. And then for the beta function, which is what we're trying to prove, we have this formula over here. And for the actual proof, we have that this, for the base step, we have the beta function equivalent to the alpha function. That means we can proceed to the inductive step, which we also found that those two, the beta function is also equivalent to the alpha function, which means that by principles of induction, the beta function holds true. So we found 3 more additional results that was in the process of proving the conjecture. So the first one was a relationship between a special case of a simple random walk and a special case of random walk generated by random permutation and simple random walk.
So for this special case, you have to start at the origin and also end at the origin. So what we did here is that we add an extra step and the slope, and collect the slope from this point to that point, so that the slope has sum of 1. And we call the distance between the vertex and the slope as elevation. And we prove that there can be only one way to rank the length of the elevation. So for example, the elevation here is the largest, so we label that as 5. And the elevation here is the smallest, so we label that as 1. So for the next two additional results, we found the divisibility power of 2 for factorials, which is n factorial is divisible by 2 raised to n minus 1 if and only if n is power of 2. And what we did here is that we simply separated it into fractional parts and found the properties within that. And for the third results, we actually use result for number 2 to prove this one. Well, for this one, it is simply that the power of 2 is held constant for the two products. What we did here is that we first found the generalization for the first product. First common order is 2 raised to m. Then we simply use induction to find that it holds true for the entire product. So in conclusion, the conjecture was not proven, but it was verified at up to n equals to 100, which we stopped at that point because of the lack of computing power. But it also appears for the conjecture to hold for all parities, which initially is only for even parities. And the Chang-Feder-Grunbaum theorem was proven using induction. For as additional results, the distribution formula for a special case of Reynolds walk is found to be true. And lastly, additional results number 2 and number 3 actually show evidence supporting the conjecture, even though you initially saw it to be against it. As future work, we could verify or find a counterexample for the conjecture, or we could investigate that up to all parities, or we could rigorously prove the conjecture for all cases, or even find any additional side results that can be related to the conjecture that might be helpful. Or we could, lastly, we could find, discover if there were any simpler proof to the Chang-Feder-Grunbaum theorem. And I would like to give my thanks to my research mentor, Dr. Starr, and my faculty advisor, Ms. Gilmore, for their advice, time, and dedication throughout the project. And here are my sources.
Good evening. My name is Mrsa Freeman, and my project is on censoring toxicity in chat logs. So today online, you can kind of say whatever you want without any restriction. And for this project, I'm talking specifically about people who can say swear words or racial slurs or just spam the keyboard and other general insulting things and degrading things to each other online. So for my project, the goal was to create a sensor and test if the method of censoring used is efficient so I can build on it in the future and use it online. So for my project, I took in a bunch of comments from Wikipedia, split them into 2 histograms of words that were messages that were considered bad and messages that were considered good, and then took the appearance of the words in each histogram and compared them to each other to create a list of censored words. And then these words in the censored list were given point values based on how they appeared in each histogram. And then we ran this censored list against more messages and to test the data, to test the censor. And the messages were given a 1 if they needed to be censored and a 0 if they were permissible. So in my first function, I took those comments from Wikipedia that had been previously manually tagged for containing toxicity and other spam kind of stuff online, and took the ones that had been tagged and histogrammed them, and the ones that had been left alone and histogrammed them to create my good list and my bad list. And then in my next function, my sensor creation function, I took the bad list and compared it to the good list based on appearance. So if a word appeared in the bad list twice as much as it did in the good list, that word would be added to the censored list. And if it appeared pretty commonly in both lists or only in the good list, then it would be treated as just a regular word like hello and left alone. Later on in this function, I assign point values to each word in the censored list that has now been created. So going back to those words from the censored list and in the list, the bad list, if it appears in the bad list more than the good list, we take the appearances of the bad list and subtract the appearances in the good list to create a point value, whereas if it appears in the bad list and is not at all in the good list, indicating that's like a swear, then it'll be given a point value of 50 right off the bat. So in my final function, we take another 50,000 messages from Wikipedia that had once again previously been tagged and run them against the censored list by splitting them up, each message, into their individual words and looking at the words and how they rank up points on the censored word list. And if their point values add up to being over 5, then they will be given a 1, meaning they need to be censored, and that's 0, meaning they are, if they don't have a point value of 5 or less, then they will be allowed in chat. So here we have an example where we have just a bunch of character spam and then an insult with a swear. So the program detects this spam and then later detects this swear word and totals up their points equaling to over 5 and giving it a 1, meaning that if this was to be implemented, it would be censored. So with these 50,000 messages I took from Wikipedia, I found that about 18.2% of them had been tagged by my program to be censored, whereas when they were manually tagged in the past, only about 4% of them had been tagged. So I had about a 14.2% increase. And the reason for this is that misspellings or conjoining words or slipping in additional characters to slip pi sensors can mess with my program because it doesn't account for that yet. And so this causes overfiltering or underfiltering, which is kind of a subjective issue because the content you see in chat is based on what you want to see. So some people don't care about what's in the chat, they don't even read it, so it doesn't matter to them. Whereas some people might have their kids browsing the internet and they don't want them to see any slurs or swear words they wouldn't want their kid to use around the house. So whether it censors more or less is kind of up to the user. So to improve upon this in the future, I would like to minimize that gap that I had in the results slide by refining the method of censoring now that I know that it can work if you histogram them and compare it. And then also to account for the preference, I would like to add a customization feature that allows you to select what you want to be censored, possibly through a kind of category checkbox kind of method where you select, like maybe you don't want to see spam or maybe you don't want to see slurs or common swears, or possibly by using a slider of strength so that at the bottom of the slider it won't be censoring anything, whereas as you go up it'll censor more and more to where the top of your slider it'll censor all slurs, all insults, all swears, and all spam. So I would like to thank my parents for their support, my faculty advisor, Mr. Sproul, for his guidance through this project, and Ms. Chen for her incredible patience. And here are my sources. Thank you.
Hello everyone, my name is Srijit, and my project is on genes regulated in diabetes and heart failure. So what is diabetes? Diabetes is a disease that affects the way your body produces or uses insulin. As we can see here on the left, over 422 million people currently have diabetes, and it's on the rise. And there are two main types of diabetes, type 1 and type 2. Type 1 diabetes being where your body doesn't produce enough insulin, and type 2 diabetes is being where your body produces the insulin but doesn't know how to use it. This can lead to other consequences such as stroke, heart attacks, kidney failures, but is most commonly associated with cardiovascular diseases. In fact, a person with diabetes is 2 to 4 times more likely to have a cardiovascular disease And as you can see here on the right, this is a huge problem because primarily in the south, there's a lot of heart failure rates. So what was the purpose of this experiment? The purpose of the experiment was to identify genes using linking these diseases from the human subset, and we wanted to test these genes in the mouse models we have in the lab. So how to identify these genes? So prior to my work in the lab had performed transcriptomatics and bioinformatics, basically using RNA-seq data from human diabetic heart failure patients, and they basically compiled a spreadsheet of all these genes. And as you can see here, by fold change and p-value, fold change being the change in expression over time, and p-value being statistical value statistical value that the expression changed over. So a p-value less than 0.05 is usually considered significant. So the genes I ended up choosing were CHIL1, PTGS2, EYA2, and ATF3. CHIL1 had a p-value of 0.02 and a fold change of 0.43. It was also trending upwards in African Americans and type 2 diabetes, which is one of the reasons I chose it. And PTGS2 had a p-value of 0.036 and a fold change of 2.896. And these two genes had previous links to cell proliferation and inflammation, which is one of the reasons I chose these genes. The other two genes, EYA2 had a fold change of 2.563 and a p-value of 0.032, and ATF3 had a fold change of 2.699 and a p-value of 0.21. These two genes had previous links to cardiac remodeling and had links to cancers. So what were the two mouse models I used? The first model being STZ, which is an antibiotic that kills pancreatic beta cells, which make insulin, and the other one being MGLUT4, which is an overexpression of the GLUT4 transporter, which is supposed to help with glucose toxicity in the heart. So since I'm not allowed to handle the mice, this is what the people in the lab did prior to my work. So the mice were treated starting around 8 to 10 weeks. The control mice were given a nitrate citrate shot, and the diabetic mice were given 5 injections of STZ. And the MGLUT4 mice was transgene induced, so basically they were given a shot of doxycycline and doxchow following that. After 4 weeks, they ended up collecting the heart, liver, spleen, lung, gastrocnemius, and tail for genotyping. For the purposes of my project, I ended up using the heart only. So with the help of the lab, I performed RNA isolation. And to do Qpcr, which is the main part of my project, I needed to change this single-stranded RNA into double-stranded DNA. So I used an enzyme called reverse transcriptase, and I changed this RNA to cDNA. After doing that, I did cyber doing Qpcr, and what cyber is a fluorescence that ends up amplifying in Qpcr. So basically, you make a master mix of the cyber, gene primers, and nucleus-free water, and you basically add the cDNA to it, and once you do that, you put it through a PCR thermocycler, and during the heating and cooling process, the bonds break and the fluorescence is released. I then did gel electrophoresis just to confirm the Qpcr product. So as you can see here, CHIL1 ended up amplifying at around 35 cycles. The more number of cycles it takes, usually the lower the expression of the gene. So we kept the cutoff at around 30 cycles here. So we can kind of assume this gene didn't have any real significance in these two mice models. And PPIA here was my control gene. This is run beside my CHIL1 to make sure that my pipetting, or if anything like the cDNA was denatured or anything like that, make sure if anything was wrong. As you can see here, it was ran correctly, it amplified around 20 cycles, and had one consistent peak. So there was no problems there. And I also ended up running gel electrophoresis on this particular gene because it did not work, and I wanted to see if it was correct. And as you can see here, in synthesis 1, there was a multitude of bands. This being due to the fact that in my first trial, the cDNA was denatured. In the second trial, you see one single band, which proves it was CHIL1, and there was nothing else to see there. The other three genes, as you can see here, PTGS2, EYA2, and ATF3. EYA2 and PTGS2 were trending upwards in STZ. They didn't claim statistical significance, but they were correct at least in the fact that it was an STZ model and they were trending upwards. ATF3 was the only interesting one due to the fact that it was downregulated in GLUT4. This was interesting because we had only expected a change in STZ, as MGLUT4 was something that we were testing on the side and not really considering. And so this particular model was done to the control gene PPIA. This one was done to the control gene beta-actin. It was run by another student who came in after me, and he ran on the same mouse tissue. So basically, as you can see here, all it proved was that ATF3 was also more downregulated in GLUT4, and PTGS2 and EYA2 were both trending upwards in STZ. So for future studies, I want to look more into ATF3, primarily doing a Western blot to check the specific proteins in ATF3. And I also want to rerun PTGS2 and EYA2 as they too had varying CT values. I would like to acknowledge my family, my lab, Dr. Wendy's lab, and my primary person that helped me, which is Luke. And I also want to thank the Alabama School of Fine Arts, Mr. Ash, Ms. Main, Mr. Ware, and Ms. Chin.
Hello, my name is Ryan Montgomery, and today I'll be talking to you about the synthesis and characterization of quercetin, tanic acid, and citric acid iron complex encoded nanoparticles. So what is the problem? As you can see here, penicillin was one of the first antibiotics to be used against staph infections. After a long and heavy use of penicillin, there came a new strain of Staphylococcus aureus. And this Staphylococcus aureus was resistant to penicillin. What that means is that it was not able to be killed as efficiently as it would have otherwise. And thus the doctors developed a stronger antibiotic. This was methicillin. After heavy use of methicillin, there came a new strain of Staphylococcus aureus, and that was Mrsa. This is a superbug. It is resistant to some of the strongest antibiotics that we have created. And thus, the doctors created an even stronger antibiotic, some of the strongest antibiotics on the market today, vancomycin. And, as predicted, in 2013, there arose a case of a new strain of Staphylococcus aureus. This Staphylococcus aureus was resistant to vancomycin, the strongest antibiotic on the market today, Mrsa. Do you see a pattern here? When the doctors discover a new strain of resistant bacteria, they create a stronger antibiotic. However, these bacteria always seem to develop resistance to even the strongest antibiotics that we can create. And so instead of creating a new, stronger antibiotic, I propose that we use flavonoid nanoparticles. You might be wondering, what is a flavonoid nanoparticle? Well, let's break it up. What's a flavonoid? A flavonoid is a naturally occurring compound with natural antibacterial abilities. The three flavonoids I'll be using are citric acid, tanic acid, and quercetin. Citric acid, as you may know, is used in oranges, quercetin is found in wine, and tannic acid is used in the tanning of leather. So what are the benefits of using flavonoids? Well, firstly, they are naturally abundant, meaning there are a large quantity of them. Secondly, they're able of attacking the bacteria in such a way that they are incapable of developing resistance as quickly as they would have been using antibiotics. And they can be created rather fast, within 2 to 3 days. So now that we know what a flavonoid is, what is a nanoparticle? A nanoparticle is an engineered particle on the nanoscale. The job of the nanoparticle is to transport the compound through the body into the bacterial cell for neutralization. There are two types of nanoparticles: coated nanoparticles and complex nanoparticles. Think of the coated nanoparticles like an iron ball, and wrapped around that iron ball are the compounds, in this case, quercetin. Think of the complex nanoparticles, like two compounds coming together, binded by the binding molecule, in this case iron, and then folding in on itself to create a fully formed nanoparticle. What are our goals and our methods of creating these nanoparticles? Depending on the type of nanoparticle, we have two different procedures. They can be seen here. And so what are we looking for? What is our goal? We're trying to create a batch of nanoparticles that are of one consistent size. The more consistent the size, the better they should be against bacteria, the higher the efficacy. And so what does that look like? Consistently sized batch of nanoparticles will have a narrow size distribution with little to no contaminants. So let's see our results. Starting with the complex nanoparticles, as you can see, the size distribution of the tannic acid is rather large, with many unincorporated nanoparticles right there. Shifting over to the quercetin, you can see the size distribution is slightly narrower, with fewer unincorporated nanoparticles. Note, when I say unincorporated nanoparticles, I mean that the nanoparticles did not fully form, like the large peaks right here. They're instead the smaller peaks. Continuing. The citric acid complex nanoparticles have an even narrower size distribution in general. However, there are many more not fully formed nanoparticles. Another thing to note, looking at the picture of the citric acid complex nanoparticle, you can see that a film has formed over some of the nanoparticles. This is due to polymerization, which I'll talk about later. Looking at the coated nanoparticles now, you can see that the size distribution of the tannic acid is much narrower than some of the complexes. However, there is one contaminant, a dust particle. Shifting over to the quercetin, we can see that the size distribution is just as narrow. However, there are no contaminants. And lastly, looking at the citric acid-coated nanoparticles, we can see that the size distribution is rather large, with many unincorporated nanoparticles. We believe this is also due to polymerization. So what is this polymerization that I've been speaking about? Polymerization is when something goes wrong within the creation of the nanoparticles at some point in the procedure. The process of the synthesis has been broken. So that happens here and from here, meaning that the nanoparticles did not fully encapsulate all of the compounds that they could have. And so what does all this data mean? Well, looking at it, we can determine a good candidate for future research. Looking at the size distributions, on average, quercetin is the compound with the most narrow, or narrowest, size distribution with little to no contaminants. Therefore, it is a good candidate for future research. In the future, we would like to create a procedure that can create nanoparticles without film, whether through using different medias, pHs, or concentrations. And we'd also like to use an infrared spectroscopy that can help further determine the quality of the nanoparticles. Think of it as an added layer of quality control. And finally, as a big picture, we would like to test the nanoparticles for their efficacy against E. coli, determine how good they are at destroying bacteria. Here are my sources and my photo credits. And lastly, I'd like to acknowledge my family for their support. Dr. Bao, my mentor, Dr. Legrone, my advisor, Ms. Chin for her help, University of Alabama Chemical Engineering Department for letting me use their labs, and finally ASFA for letting me give this talk. Thank you.
Sound check. Excellent. Alright, good evening everyone. My name is Toby. I am a four-year senior here at the Alabama School of Fine Arts, and it is my pleasure tonight to present to you my research on designing and prototyping a 6-degree of freedom telepresence robotic arm. So, what is a telepresence robot? Telepresence robotics refers to the set of technologies pertaining to controlling and operating unmanned robotic devices in environments separate from the one that the human pilot is in. An excellent example of that would be this drone, which is very common in today's world. These devices are typically used in exploration, as they give humans access to areas and places that normally would not be deemed accessible due to things like inordinate cost or dangers like lack of oxygen, toxic gas, high pressures, radioactive materials, etc., etc. To that end, these devices are often equipped with articulated arms and camera systems to allow the pilot to interact with their environment should the need arise. And here I have a picture of a submersible vehicle that is equipped with both the camera system and the robotic arms that I just mentioned. So what is the purpose of this project itself? Well, a lot of the arms that you see these devices equipped with today are they're very limited. They're limited only to rigid, well-defined, pre-programmed motion paths and aren't true free-moving arms. So they're very limited in the things that they can do and the applications that they can perform. So the goal of this project is to create a fully functioning 6-degree of freedom arm capable of fully mimicking the motions of a human arm. Anything you can do with your arm, you should expect this arm to be able to do as well. To that end, the materials I'm using are the Highwonder X-Arm2S, which is a pre-built robotic arm kit. The reason for that is the lab that I did my research in did not have the time, money, materials, and I did not have the skill necessary to machine my own arm. I'm using an Arduino Uno as a heavy lifter for most of the calculations involved in this project. I'm using two MPU 6050 6-axis inertial measurement units to control to obtain data from both the wrist and shoulder joints. I'm using a sliding potentiometer to control the actuation of the claw gripper, and I'm using a rotary potentiometer to encode the position of your elbow, jumper wires to connect everything together, and here is a wiring diagram for one of the MPU 6050s. As far as construction of the arm itself goes, I first started by assembling the arm, establishing serial communication between the Arduino and the arm, connecting the sensors to the Arduino, and establishing communication between all three. In order to control the arm, we first start with this feedback loop here. We start with movement of the pilot's arm. There are sensors connected, the sensors connected to your arm constantly read the data from, or the Arduino constantly reads data from the sensors hundreds of thousands of times per second and updates the projected final position of the arm. But the issue with that is this is far, far, far too fast for the Arduino to actually be able to send this information to the robotic arm and tell it to actually move. The information would just get overwritten before it was actually sent, and that would cause a lot of issues. So the Arduino, after a set amount of time, will trigger what's called a hardware interrupt, which basically just pauses that feedback loop and tells the Arduino to compile that data into servo commands and send those servo commands to the actual arm so it can move into its final position. Here are a couple of images. This is the overall setup of the arm itself. Here I have a close-up view of the microcontroller and the Arduino, along with the serial communication and wiring for all of the sensors. And here is a picture of the arm itself. As for my results, did the arm work? Yes, it did. It's very fast and very responsive. I believe the final refresh rate came down to about 10 to 15 times a second. I couldn't tell you the actual amount because all of these times are measured in microseconds, but that should be the range that it falls in. And it's relatively accurate, barring a couple of conditions. So due to an issue in sensor calibration, the arm tends to overshoot its projected final position near the extremes of its range of motion. Anywhere that you would have to stretch to get to, the arm will struggle to copy. Now what does that mean? Consider these two figures here. The black one is the human operator, and the blue one is the robot. Right now they're in their calibration configurations, but let's say the human pilot raises his arm to this configuration. What we would expect to see the robot do is copy that position exactly, but what actually ends up happening is the robot will over-rotate its wrist joint, resulting in an overshooting of its final correct position. And the reason for that lies in the two IMUs, one on the shoulder and one on the back of the wrist. Since both of these are calibrated relative to the horizontal, when the user and the pilot raises their arm to this position, this IMU is returning an angle of roughly 90 degrees to the horizontal, and this IMU is also returning an angle of roughly 90 degrees to the horizontal. However, you'll notice that when the pilot raises his arm, the wrist joint itself doesn't actually rotate. So the wrist IMU should be returning an angle of 0 degrees, but it returns an angle of 90 degrees, which is what causes the robot to rotate not only its shoulder joint, but also the wrist joint. So it's not wrong, but it is wrong, and that's not good. So there's also a slight issue with sensor noise. So the two potentiometers that I mentioned earlier are a tiny bit noisy in that they don't give me flat values. What we expect to see from a potentiometer, so I'll use these as an example. I give these potentiometers a flat 5-volt input as a power source. And then depending on what I set them to, I should expect to see a flat voltage returned. So let's say I open the sliding potentiometer to 50%. I would expect a voltage of 2.5 volts returned, but in reality it fluctuates between about 2.3 and 2.7 volts. So that causes the robot to jitter around a little bit, and it's really shaky, almost like a sleep-deprived high school student who had too much coffee that morning. So in order to fix these issues, I would like to switch to using inverse kinematics or position control instead of using a sensor fusion. And the reason for that is inverse kinematics is far faster and far more precise than a sensor fusion is. To understand inverse kinematics, we first have to understand forward kinematics and kinematics in general. In robotics, the study of kinematics is the study of a robot's joints and its end effector. In forward kinematics, I'm given a list of joint positions, and I can use those to calculate the final position of the end effector in 3D space. So then in inverse kinematics, I'm given the final position of the end effector in 3D space, and I can use that to calculate the joint positions that I need to set them to in order to get the end effector to that final spot. I would like to correct, if I can't do that, I would like to correct the sensor calibration issues, and I would also like to fix the noisy sensor output by applying something like a moving average filter to the sensor data. I would like to thank Dr. Shen Xianrong in the Department of Mechanical Engineering at UA for allowing me to use his lab. I would like to thank my faculty advisor, Mr. Kwon. I'd like to thank Ms. Chin and all of my friends and family here at ASFA and back at home who have allowed me to get to this point so far. Thank you.
All right, hello everyone. We're nearing the close. My name is James Shi, and I'm a 5-year senior at the Alabama School of Fine Arts. And for the past year, I've been working in a biochemistry lab on determining the best small hairpin RNA to knock down the Atrx mRNA in stem cells. So a little bit more about what my research is in. So my research is in the field of gliomas. Gliomas are cancer cells that originate from glial cells. Two examples of glial cells are astrocytes and oligodendrocytes, as you can see in the diagram, blue and green. And these are in the brain, they're between neurons, and they help the neurons carry out their functions. And these cancer cells make up around 1/3 of all brain tumors, with the other 2 coming from other cells in the brain. And also, an important thing to look at here is the cancer cells aren't necessarily on its own. It's not like you have a plastic ball in your brain. They're mixed in with the brain matter, which, as I will discuss on the next slide, makes it very difficult to actually surgically remove all of it. So currently, how do we diagnose it? So first you experience symptoms like headaches or seizures, and depending on which lobe of the brain the tumor is located in, you might have lobe-dependent symptoms as well. So like if it's in your frontal lobe, you might have judgment issues. And then you go in for treatment. They first do a surgery. They can't remove all of it, like I said earlier, since it's mixed in with brain matter. pros versus cons, you don't want a third of your brain removed. So they remove majority of it, they do a biopsy on it, and if it is a glioma, then they go ahead and give you radiation therapy or chemotherapy to kind of slow down its growth, but it can't cure it. So right now, it's impacting around 700,000 people, and this year alone, it's projected that 90,000 more people get diagnosed with it. But don't worry, not all these people have malignant tumors. Some of them have benign tumors, so the numbers aren't as bad as you think. Well, they're still pretty bad. So in this diagram here, you have a patient in 2007 with a low-grade glioma. So this is like a grade maybe 1 or 2 glioma, pretty small. You could barely even notice it, honestly. And then 2014, boom, it's really big. It's like a fourth of the brain. So this is what we call progression, and this is from grades 1 to 4. So this is the most extreme cases on both ends. The World Health Organization classifies it from grades 1 through 4. 1 through 3 we know as lower grade gliomas, and grade 4 we call glioblastoma or a high-grade glioma. In this diagram here, we have a secondary glioblastoma, which means that it's progressed there. If the patient comes in and is immediately diagnosed with a glioblastoma, we call that primary glioblastoma. In my lab, we'll be working with secondary glioblastoma and specifically the lower grade gliomas. So, as I mentioned earlier, the two different types of glial cells can form astrocytoma and oligodendroglioma. There's also more like a mixed type as well. But for the purposes of this study, I'm going to focus on astrocytoma because oligodendroglioma has a co-deletion of chromosomes, which would be a little bit hard to mimic in a lab. So focusing on astrocytoma, there are 3 different mutations. There's the IDH1 mutation, which converts the alpha-ketoglutarate glutarate we need in our Krebs cycle to 2-hydroxyglutarate, something we don't need that much. And then also P53 gene, it's a recognized tumor suppressant, so without it, cancer cells are more likely to grow. And finally, I'm going to be focusing on the Atrx gene. We don't actually know what it does, actually. So scientists think that it might have something to do with alternative lengthening telomeres, which means every time DNA is replicated, the caps don't get smaller, so the cell never commits apoptosis, making cancer cells immortal, or something to do with histone chaperones. And this is why we need a research model. So a research model will allow us to test more innovative treatments and also find out what exactly stuff like Atrx does. So as overall, the research goal is to combine the Atrx, or the mimicking of the Atrx mutation, with the mimicking of the IDH1 and P53 mutations. My specific goal is to find out which of 5 different Atrx kind of like vectors to attack the Atrx mRNA, which one works the best. So this is our vector. So a few things to point out on this vector. First, the ampicillin resistance down here. So whenever this is in our bacteria, in our E. coli, only the E. coli with this will survive and be selectively bred when we amplify our plasmid. Secondly, the long terminal repeating end. So right here, this tells you that only the top half will actually get transcribed in a cell, much like how DNA in your nucleus is transcribed. Thirdly, the desired small hairpin RNA. The small hairpin RNA is right here. It's going to form a small hairpin. In the sense of DNA, it's palindromic, so the front and the back ends are complementary and they attract each other to form the small hairpin. This ultimately will go in the cell and interact with something called a RISC complex and then ultimately destroy the Atrx mRNA by cutting it. So I was given 5 of these that we purchased from a vendor, each of them having an shRNA that targeted a different site for a total of 5 sites. So first, our lab is, like most labs, funded and has a budget. So I didn't get 200 milliliters of plasmid like I needed. I got 10 milliliters, so I need to first amplify it. So what I did was I put it inside bacteria through heat shock transformation. Then I grew it over 2 nights and then purified the plasmid. And finally, I tested that it was indeed plasmid and its concentration was high. This is gel electrophoresis, just like some of my fellow classmates did. So the small bands here are when the plasmid was cut by restriction enzymes, and these bands are good actually for plasmid, so we know it's plasmid. But here we have low plasmid concentration, ended up redoing this one. High plasmid concentration, we kept it and we used it for the next step, which involved putting inside a retrovirus. So the origins of this come from the HIV virus. When scientists were studying it, they decided, okay, what if we can take out the actual detrimental effects of HIV and instead put in genome of our choice instead of the actual HIV genome. So that's what I did here. I put in my plasmid inside the virus, and then once I got the virus, I transduced it into new cells, so I infected new cells. And now that the new cells have been infected, what I can do is I can purify the RNA because I want to see which one of the 5 knocked down the most Atrx mRNA. After I purify, I turn it into cDNA, and then I ran Qpcr on the test to kind of quantify the amount of Atrx mRNA that are in the cells. So the intermediate result was the plasma concentration. That was the first half of my project. So I had to get a certain level in order to make the virus. Next, the Qpcr test. The raw data I did not show, did not show here, but this is the normalized data. So first, I normalize it to beta-actin, the housekeeping gene of my choice here, and then I normalize it to the control, which you may have noticed already is at 100. So what this graph shows is, say the control, it knocked down 0% of the Atrx mRNA. So control was just a random small hairpin, does nothing to cell, nothing to Atrx. And then the small hairpin RNA 1 through 5 knocked down around 35, 85, etc. So our initial goal was to find out which one knocked down the most. So shRNAs 2 and 3 had the least remaining, so they knocked down around 85% each, so we'll probably use those in future experiments. We don't actually care which one, we don't care if they're statistically significant or not. It's more just seeing which one looks like it knocked down the most. So we're going to choose one of these, and then also get the viruses that mimic the IDH1 and P53 mutations. We're going to take all 3 and then infect a neuro stem cell line, which is a stem cell line that comes from neuro cells. Earlier, we used human embryonic kidney cells just because they're easily grown in the lab, they're cheap, they're reliable. But when we actually want to make an in vivo model in mice, we need to use a neuro stem cell line. That being said, we're going to put it into mice and then surgically remove the tumor after a few weeks and then test it on several scales, 3 of which I listed here. So some scales would be where it's located, how long it took to grow, whether it's predictive of the patient's reaction to treatment, other characteristics, so anything like that. So if it works out, great. Then pharmaceutical companies have something to test the new drugs on. Yeah, I'd like to thank the following people for their support. Dr. Rizal, he was the PI. Dr. Luzon, she was the tech who helped me with many of my experiments. Alex is the PhD student in my lab. And Mr. Ash was my advisor. These are my references.
Hello everyone, I'm Ethan Forrest, and this is my presentation on the application of word tokenization and spelling correction algorithms. If you don't know what that means, don't worry, I don't either, but hopefully we'll both figure it out by the end of this. So, what is a spelling correction algorithm? That's a really long name, so we're just going to call it spell check. Merriam-Webster defines spell check as, that's a lot of text, you all know what spell check is. Basically, it checks a word that you type against the dictionary, and if it's not found in the dictionary, you get an angry red line, and then you fix it. There's also context-sensitive spell checks that the word can be spelled correctly, but it's not used correctly in the sentence, but we're not focusing on that today. A brief history of spell check. BCE, that's before computer era. Back then, they used these arcane contractions called dictionaries to see if your word is spelled correctly. And then in 1938, the first programmable computer is created, and 42 years later, the first PC spell checkers appear. These were standalone applications that would run within the word processing software, and they were simple, unreliable, and honestly terrible. 10 years later, Microsoft changes the game with autocorrect. This would automatically run with when you in real time while you were typing and would recommend the best word for for what you spell. It only took 5 more years for this to be adapted to mobile devices, and fast forward a few years, we get to modern spell checkers that you probably use on a daily basis. But this is not the limit. There are still many ways that spell checkers can be improved. I'm sure you've all had frustrations with spell checkers in the time that you've been using them. So, first we're going to give a brief rundown of the plan for the algorithm. The first thing that we need to do is train the algorithm. To do that, we need data. So, a word is generated. For this instance, we'll use souvenir. Then this word is given to this magic misspelling machine, which is going to give us a realistic misspelling of the word because I don't have time to sit there and make them myself. So then we get this word and we give it to our super awesome computer algorithm, which is going to tell us whether or not the word is spelled correctly. And hopefully that is right. And then it all goes again. All right, so misspelling magic machine is a little bit vague, so I'm going to explain how it works. So we have our random word, and one of 3 different algorithms is applied to this word to give us what I hope is an emulation of human error. So first we have the insertion misspell, which inserts a random letter into the word. Here R is inserted. Then we have the deletion, which just deletes a random letter from the word. Here P is deleted. And we have replacement, which replaces a random letter in the word with another letter. Here, I is replaced with O. So, it's important to keep in mind that these algorithms are trying to emulate human error, so all of the letters being changed are based on proximity on the keyboard. You're much more likely to accidentally click a key that's close to the key that you meant to click. For instance, if you meant to click G, it's much more likely that you click these letters, and then it's slightly likely that you accidentally click these. And the algorithm emulates that when it's changing the letters. Now, we get to the word tokenization part. So what is a token? Basically, tokenization is just counting the number of appearances of each letter in the word. So for instance, here is the tokenization for the correctly spelled word, and the insertion misspell, deletion, and replacement. So humans are not very good at telling these tokens apart. If I asked you if one of those that I just showed you was wrong, you would have no idea. All right, so this is the super awesome computer algorithm. It lives up to its name, huh? This is a CNN, not the news channel. It's a convolutional neural network. So this one is pretty complicated, so I'll simplify it down. These circles you see are called nodes. They are weighted based on they have weights, and these weights are basically just multipliers on a line that the model will plot whatever word we give it on top of. The vertical position of the nodes indicates the weight. So when we give the word an input, it's going to pass the word through these nodes and give us an output. If the output is right, we're going to leave it alone. If it ain't broke, don't fix it. But in the very rare case that the algorithm is wrong, then we're going to adjust the nodes so that that doesn't happen again. Now, the nodes moved a lot just now. If we actually did it, they wouldn't move very much, because if we move them that much every time that it gets it wrong, it gets into this really specific configuration where it only works for words it's seen before. And we don't want that because English is changing very quickly, especially with the internet. We get new words every day. So remember, that was a simplification. There are actually hundreds or thousands of these nodes with several different layers that all work together to reach the desired output. So I've explained to you how the algorithm works. So now we find out, does it work? Kind of. And I'll explain why so that you don't blame my algorithm for it not working, because that's not the problem. So the computer sees data that looks like this. Now, if you saw this, you would have a hard time telling me what that word even is, much less if it's spelled correctly. And now everybody here is thinking, oh, computers are smart, surely computers could figure it out. Apparently not, at least not consistently. Also, we have some instances like this, where we get 2 words that we can clearly see the first word is spelled correctly, it's a valid word, but the second one is not. And to us, that's obvious, but what the computer sees looks like this. And they're identical. And understandably, this can be a little bit confusing for the model when it's trying to figure out whether or not the words are spelled correctly or not. Now, when I say it doesn't work or doesn't work well, it's a little bit misleading. It's still about twice as accurate as guessing randomly, but it's nowhere near the original goal that I'd set. So, looking towards the future, the most important thing for me is just to make it work. And I'd like to do this by reconfiguring the input so that the model has a little bit more information and isn't just working off just the letters. And also changing the number of nodes or potentially changing the type of regression that the model uses. That's all for my presentation. Thank you.
Thank you, Toby. Can you come on down, Toby? Okay, these seniors have done wonderful the whole day, and they are very, very tired. So can we have all of them up on the stage, please? Come on up. Toby, you're on your way down, right? Here's our group of 15 seniors. We actually have 16, but I think Lizzie is not feeling too good. She's running a fever. So I want to make sure you'll see them, and we're very proud of them. They worked so hard. Wonderful job today. Let's give them a big round of applause. The next thing I would like to do is actually recognize a few guests. Dr. Mitchell, our president of ASFA, thank you for coming. Beth Mulvey, director of development. I'm not sure where you are at, but thank you for coming. And also, this is the first year that we have so many alums that came back to visit and support this symposium. So the very first one I want to introduce will be Dr. Carl Lancaster. Please stand up. Way back there, I think. Dr. Azim Marson, class of 2014. Thank you for coming. Catherine Wilson, where are you? Class of 2019, and two more, Ricardo Crenshaw and Sam Borkin, class of 2020. Thank you for coming. And I know everyone is hungry, and we have a big reception here. Please join us and then mingle around and do socialize with our group of seniors. Thank you.